We are now ready to define profile hidden Markov models for classifying proteins into family, a difficult task in bioinformatics. Classifying proteins into family is important because as soon as a protein is classified as a family member, we know the function of these proteins. However, the similarities may be subtle for diverged protein, and therefore pairwise similarities may fail to detect what family a given newly sequenced protein belongs to. However, protein may have weak similarities with many proteins in the family, and it will help us to assign this protein to a family, but we need new computational techniques for finding such subtle similarities. Let's start from the alignment of a number of proteins as shown on this slide, and let's remove columns if the fraction of insertion exceeds the threshold theta, the maximum fraction of insertions per column. And after we've done this, we'll construct alignment when each column has relatively small number of insertions. We can further construct profile of this alignment, and finally we will construct an HMM corresponding to this profile. This is actually a strange HMM because the probability of transition from every state to next state next to the right state is 1. Therefore, there is nothing probabilistic about this HMM with respect to transition probability matrix. However, with respect to emission probabilities, there is a probabilistic meaning of this HMM, and it corresponds to every new protein having certain probabilities being emitted by this HMM. But this HMM has a major flaw. It is not clear how to model insertions in this HMM. For example, how would we represent this protein in the framework of this HMM? Well, an obvious way is to add insertion states like this one, and if there are multiple insertions, we will also connect insertion state with itself, and finally we will design this HMM of this insertion state. Now we took care of insertions, and this would be paths in the HMM corresponding to this protein, but what about deletions? And there is a simple way to account for deletion, let's say for this deletion, by introducing transitions between all states to the right in this HMM diagram, like this state. However, the question arises, how many such edges in HMM diagram we will have after adding all these possible transitions? In this case, we added only transition for a given node of the HMM diagram, and there are already many edges. And as you remember, the running time of the Viterbi algorithm equals to the number of edges in the HMM diagram multiplied by the number of emitted symbols, so the solving the corresponding decoding problem will be time-consuming. However, there is a better way. We can introduce deletion states in the HMM diagram, and in this case we can route the sequence through these newly introduced deletion states. We need to introduce all of them, of course, and that's now how the deletion issue will be addressed with introducing relatively small number of new edges in the HMM diagram. So here's the path corresponding to this protein that is shown at the bottom of this diagram. And I want to ask you a question, are there any edges missing in this HMM diagram? And of course, there are missing edges between deletion and insertion states. We will add them, and our HMM diagram is almost done. The only thing we need to add is the start and end states. However, the question that remains unanswered is what are the transitions and emission probabilities in this for this HMM. And we wanted to define profile HMM problem, which is to construct a profile HMM from a multiply alignment The input to this problem is a multiply alignment and a threshold theta, the maximum fraction of insertions per column, and the output, transition and emission matrices of the profile HMM. Let's construct hidden passes for every row of the multiply alignment in the profile HMM. Please note that we are constructing hidden passes in the HMM diagram, not in the Viterbi graph of the HMM. We haven't even constructed the Viterbi graph yet. So for the top row in the multiply alignment, this will be our green pass through the HMM diagram. This is the red pass corresponding to the second row, and this is yet another pass corresponding to the third row. And here, all 5 passes corresponding to each row of the multiply alignment, and the question arises, how do we compute transition probabilities based on these passes? Let's take a look at one particular node in these HMM diagrams and in all these passes. Only 4 out of 5 passes pass through this node, and rather for a 4 transition from this state M5. 3 of them go to the state I5, 1 go to the state M6, and 0 go to the state D6. And therefore, it makes sense to define transition probabilities from M5 to I5 as 3 over 4, transition probability from M5 to M6 as 1 over 4, and transition probability from M5 to D6 as 0. It turned out that this common sense assigning of transition probability actually correspond to optimal assigning of transition probabilities in the HMM diagram. But what about the emission probability? In this case, we look at the node M2, and it is traversed by 4 out of 5 passes, and the emitted symbol are C, F, C, and D from this state, and therefore we define the emission probabilities correspondingly according to the count of the emitted symbols from this state. We are almost done with defining the profile HMM for multiply alignment, but let's consider a small complication caused by forbidden transitions in the HMM diagrams. Please note that most of transitions between states in the profile HMM are forbidden, and the graph of the HMM diagram shown here only shows allowed transition. So the transition matrix of transition probability for these HMM diagrams is mainly empty. We show gray cells here corresponding to allowed transition and clear cells corresponding to forbidden transition. Everything is 0 in the forbidden transition. But please note that there are also many zeros in the gray cells. And you should worry about the 0 because they simply indicate that the number of rows in our multiply alignment is small. And therefore, don't forget about pseudo count in this HMM by adding small pseudo count for every gray cell in the matrix of transition probability for profile HMM, as usually we've done in the section when we considered finding motifs in DNA.